Hemostasis. What do you mean by hemostasis? Hemostasis is the process in which bleeding of an injured blood vessel is stopped. This prevents excessive loss of blood following an injury. Hemostasis can be broken down into two phases. These phases include primary hemostasis and secondary hemostasis. Primary hemostasis involves the formation of a platelet plug. On the other hand, secondary hemostasis involves the coagulation process and the formation of a blood clot. Functionally, hemostasis is comprised of various hemostasis stages after a vessel injury, such as blood vessel spasm, platelet plug formation, coagulation process and blood clot formation. Vascular spasm. A blood vessel experiences a vascular spasm within 30 minutes following its injury or trauma. Vascular spasm means sudden and shortened tightening of the vessels caused by its smooth muscles. Blood vessels should constrict in order to save and reduce the blood gushing out from the blood vessel. How do smooth muscle knows when to contract? Well, there are many ways. Firstly, cells around the injured area begins to secrete signals telling the smooth muscle to contract. Secondly, our own nerve reflexes will cause the smooth muscle to contract. This leads to vasoconstriction, narrowing of the blood vessels, which limits the blood flow to the injured area by blocking small blood vessels. In addition, the injury causes the lining of the blood vessels to expose their collagen to the blood cells. Collagen is a structural protein of a blood vessel wall. The platelets attached to this collagen get activated and secrete vasoconstrictor substances that immediately constricts the blood vessel. Platelet plug formation involves the adhesion and aggregation of activated platelets at the injury site. Firstly, platelets bind to the exposed collagen, and this process is aided by the von Willebrand factor, VWF. Von Willebrand factor is a plasma glycoprotein that helps the flowing platelets to bind to the walls of blood vessels. This binding leads to the activation of the platelets. Next, the activated platelets release granules, which contain several factors, some of which enhance coagulation, blood becoming thicker, almost like gel. Some factors released from platelet granules include thromboxane A2, serotonin, adenosine phosphate, ADP, and epinephrine. Adenosine phosphate, he is the one who will recruit more platelets to the injured site. In addition to releasing granules, activated platelets also increase the expression of their surface proteins, such as the glycoprotein complex. This complex binds to a soluble plasma protein known as fibrinogen. He is the one who cross-links between numerous platelets. Ultimately, the injured site gets dominated by layers of activated platelets that eventually form a platelet plug. A platelet plug closes the opening of the blood vessel wall, thus reducing further blood loss. Almost done. But this plug is temporary, it is not that strong. So further processes are required to strengthen this plug, such as clotting factors. This plug is then strengthened by fibrin, a strong and insoluble polymer that forms a mesh intended to trap circulating blood cells. This step we'll discuss in the next video. Bye.